we're just going to jump straight in. So, immunotherapy, what exactly is it? Just to go over immunotherapy in brief. So I'm sure we don't have to introduce cancer to this group. But generally speaking, immunotherapy is sort of any treatment that helps your body's immune system fight cancer. As we're aware, your body has innate defenses that are sort of built in to protect you against, foreign threats, pathogens, etc. And this takes the form of the immune system. And so the concept behind immunotherapy is immunotherapy strategizes to better equip your immune system to fight and kill cancer, as opposed to using exogenous agents like chemotherapeutics and or radiation. And so just on a philosophical level, immunotherapy aims to equip immune cells with better weapons, better intel, and better direction. So more concretely, the exact strategies and modalities of immunotherapy that are either approved or have some exploratory preclinical work done on them include these 5 listed here, which I pulled from cancer.gov. So just briefly, they include immune checkpoint inhibitors, T cell transfer therapies, monoclonal antibodies, vaccines, and immune system modulators. And I'm actually going to talk about these in reverse order because we're going to spend a little more time in the last one. So immune system modulators is really the most broad-scoping general bucket of immunotherapies and really can include almost anything that doesn't fall into the other categories. But like the most classical example would be IL-2 therapy, which is interleukin-2. The classical function of IL-2 is to act as quote-unquote T cell food, such that you need IL-2 for T cells to proliferate and mount an effective immune response. Much in the same vein, we have vaccines where you sort of challenge the immune system with an inactivated or a chunk of the threat that's non-infectious and non-pathogenic. So just to sort of prime your body against, a re-challenge of that same particular pathogen, or in this case, cancer. So in the case of cancer, the antigen you would prime with are these things called tumor-associated antigens, among other things. Jumping up to monoclonal antibodies, we know what those are. They're produced by the body. They have very specific and very high affinity for their particular ligand, and they typically bind to these things to neutralize them and prevent them from exerting their function. In many cases, for example, you can think of the viral spike protein. If you have antibodies bound to those, it prevents the virus from entering the cell. And it can also mark things to be phagocytosed by cells such as macrophages. So in the case of cancer, you might have antibodies against cancer cell surface proteins that then mark them for phagocytosis. T cell transfer therapies are actually one of the most exciting in my mind, and they really gained a lot of traction in the past 5 years or so with the approval of the first 2 autologous T cell transfer therapies, Kymriah and Yescarta, I believe, from Novartis and Gilead. The concept behind these is to remove a patient's T cells, engineer them. So hold on, I gotta kill an ant that I just saw. Okay. Yeah, to remove the patient's T cells, engineer them and expand them in vivo such that they express a chimeric receptor. We won't go into too much detail of how that works, but once those super efficient T cells are cloned and expanded, they're transferred back into the patient, and those then go on to exert cancer cell killing effects. And generally speaking, they're quite good for blood-based cancers, not so much for solid tumors, which is still under investigation. And lastly, all the way back to the beginning, with immune checkpoint inhibitors, these are probably the best studied among all of these except for monoclonal antibodies, and they really gained traction in the last decade or so. And the work in this particular field won the Nobel 2015 prize in medicine. So how they work is, generally speaking, your immune system has the ability to recognize and respond to foreign bodies, but it also needs a way to taper those effects to prevent your body to prevent like alloreactivity with your body, prevent your immune system from damaging your own body. And so the pathways it does that, it accomplishes this is through immune checkpoints, which is molecules and surface proteins that when they bind to T cells, for example, it'll cause immunosuppression and deactivation and exhaustion of those T cells, and essentially prevent them from continuing to be cytotoxic and destructive. And so we're just going to talk about two of the best studied ligands or best studied immune checkpoints today, CTLA-4, which is a cytotoxic T lymphocyte-associated protein 4, and PD-1, which is programmed death protein 1, and its associated ligand, which is PD-L1, programmed death ligand 1. And we're going to touch on a couple of the others, but we're not going to go too into depth. Great. So before we talk too much about the immune checkpoint inhibitors, we need to understand a little bit about T cell biology and the T cell lineages. So just in broad strokes, there are two main T cell lineages, those being CD4 positive and CD8 positive T cells. And each of those T cell populations then have subpopulations that they can differentiate into. So can I make this a laser pointer? Here we go. So we have naive CD4 positive T cells that can then differentiate into immunostimulatory and immunosuppressive phenotypes. So, for example, they can differentiate into Th1, Th2, and Th17 phenotypes, which are immunostimulatory and are good for clearing virally infected cells, bacteria, helminths, as well as some tumor cells. But there are also CD4 cells that can differentiate into T regulatory cells, which actually exert immunosuppressive functions. And so for the purposes of this talk, I really wanted to bring your attention to the fact that this population exists and that they're derived from CD4 T cells. So the takeaway is that there are T cells that are immunosuppressive, and not all of them contribute to an increased immune response. And in fact, some of them are responsible for directly downregulating that. Now, if we step over into the other branch of the T cell lineage with the CD8 positive T cells, it's quite complex in how the CD8 T cells become cytotoxic and memory T cells. But what we're going to focus on is highlighted here in this red box, where we have, again, naive CD8 T cells that have really not been primed for anything. And then once they're presented or once they encounter their cognate antigen, they differentiate into effector CD8 T cells. And this T cell population is actually the cytotoxic T cell population, which can recognize their cognate antigen and then release perforin and granzymes in response to lyse the virally infected cell, for example. And then once the infection or the pathogen is cleared, it can then become a memory T cell. And these are sort of just hang around in the reservoir until they're re-challenged with the same particular pathogen, after which they can then mount a rapid and very much more effective immune response. Okay, now we're going to jump into naive CD8 T cells, which is what we're going to spend the majority of the talk on, because naive CD8 T cells are where immune checkpoints really have the greatest effect. So, to understand how these naive CD8 T cells become activated and how they, begin the process of becoming cytotoxic and mounting that immune response. Again, speaking in broad strokes here, you have antigen-presenting cell, which would be a macrophage, dendritic cell, or even a virally infected cell, and cancer cells that have tumor-associated antigens on their surface. All of these have MHC1 on their cell surface, which is a major histocompatibility complex. And then those cells will then express, in the case of dendritic cells, they'll move antigen to their surface with the MHC1, and they'll then present that on their surface. And once they encounter the naive CD8 T cell with the proper T cell receptor, that causes the signaling cascade that turns this, starts this naive CD8 T cell on this slippery slope of becoming differentiated, becoming an effector T cell and proliferating. So, for example, once a naive CD8 T cell becomes activated, it can go on to proliferate or differentiate. But this whole process is contingent upon a whole suite of co-inhibitory and co-stimulatory signals. The redundancy being needed to prevent, accidental activation or false positive activation, as well as, control over the dynamics and the scale of that actual immune response. So immune checkpoints actually play directly into this co-inhibitory signal suite. So before we can actually talk about the co-inhibitory suite, we need to talk about the co-stimulatory signaling suite. So in addition to this MHC and T cell receptor complex with the antigen that I talked about on the last slide, there's a co-stimulatory signal along the CD80, 86, and CD28 axis. So all naive Cda T cells have this surface protein called CD28, which acts as a receptor. And its ligand is CD80 or CD86, which is typically on the antigen presenting cell. And so this CD28, CD80, 86 complex is actually necessary in order for this T cell receptor mediated signal transduction to take effect. And so you can't have one without the other, essentially. And this actually ties directly into the first immune checkpoint I wanted to talk about. JK, we're going to back up. I forgot Fox was here. Apologies. So one thing we need to talk about is once the signal, once the co-stimulatory signal and the T cell receptor are both bound to by the cognate antigen or cognate ligand, the naive CD8 T cell begins differentiation, but it also begins surface expression of CTLA-4, which is the first immune checkpoint I wanted to talk about. So this is why we needed to talk about the CD28 receptor first, is that signaling through CD28 causes upregulation of CTLA-4 in the cell surface body, and then CTLA-4 will then directly compete with CD28 for the exact same ligands. And so by doing so, it'll decrease the frequency of that reaction or interaction, and thus decrease the compounded stimulatory signal towards the CD8 T cell. But in addition to that sort of direct competition, it can also directly signal intracellularly and inhibit this CD8 T cell activation. You'll notice I'm glossing over a lot of JAK-STAT pathways here, but we will not talk about that in this talk. And so there are really multiple mechanisms of CTLA-4 mediated immunosuppression. So one being direct competition, one being signaling intracellularly, and there's also some work being done that shows it can remove CD80, 86 from the surface of the antigen presenting cell, which is interesting to me, and I haven't looked too much into it, but I'm just imagining it binds tightly and rips it out of the membrane, which I thought was interesting enough to warrant inclusion. But again, that's still sort of an open question as to whether or not that mechanism actually, or how it actually works. Now, if we jump back into the CD4 lineage, if we remember way back in the beginning, I mentioned there are T regulatory cells that can actually mediate immunosuppression. Turns out these T regulatory cells also express CTLA-4, but in contrast to the CD8, cytotoxic T cell lineage, T regulatory cells are actually activated by CTLA-4. And so when you have signaling through CTLA-4, you have increased immunosuppressive activity through T regulatory cells. And again, this is in broad strokes, so I'm not going to talk about all the secreted factors here. Well, maybe I will. One of them is TGF-beta, and I don't remember the others. But there are, secreted factors and increased surface proteins that mediate immunosuppression and prevent, or at least attenuate, immune response. And why this is important is that solid tumors in particular, can actually co-opt T regulatory cells and recruit them directly into their mass, and sort of have them as a shield against immune clearance, essentially. And so I thought this was interesting because for solid tumors in particular, you think of them as sort of, we know cancer cells or tumors in particular aren't really homogeneous, and we know there's significant heterogeneity between tumor cells even within the same tumor. but it's interesting because there are also, hijacked cells that belong to hijacked healthy cells, and that can actually constitute up to 50% of the tumor mass. And so, by mass, the typical solid tumor isn't even entirely or even mostly tumor cell. In some cases, like mesenchymal-derived stromal cells can make up to 50% of a tumor mass, which is just insane to me. The main point being that there are many, many, many other cells beyond just the tumor cell type that are present within the tumor. And in many cases, they're recruited for the purpose of immunosuppression and suppressing that T cell response. Okay, so we now know how CTLA-4 works and how it mediates both CD8 immunosuppression and T regulatory cell immunosuppression. And it stands to reason that targeting this axis would be a fairly effective way to amplify the immune response to cancers. And so this pathway has been explored and approved. The first CTLA-4 antibody, and I believe the only CTLA-4 antibody, ipilimumab, was approved by the FDA in 2010. And it's the first therapy with demonstrated survival benefit in metastatic melanoma. So there are some therapies like that's probably not how you say it. But there are other therapies that have been approved for metastatic melanoma, but those are typically on the basis of increased response rates, so a greater subset of melanoma patients respond to it, but they don't necessarily get more survival benefit from it than older or more conventional therapies. So ipilimumab was sort of landmark in that regard, in that it had a median survival benefit of, I think it was 3.5 months, which again is incremental, but still significant for such a high lethality disease, high mortality disease. And another important note I thought would be interesting for some more clinically oriented audience is that the treatment actually takes several months to show effect. And this is true of all immune checkpoint blockade therapies, not just CTLA-4 therapies. So in contrast to conventional chemotherapies like cisplatin or 5-FU, which can take days or weeks to take effect, immunotherapies take a long time. And it sort of makes sense if you think about the amount of signal transduction and clonal expansion that has to take place in order for the tumor to shrink. But it sort of complicates things in the clinic, because during the time that it takes for the therapy to take effect, the tumor is not just sitting there, right? The tumor can actually increase in size. And so this throws a wrench in how clinicians typically evaluate, cancer response to treatment, which is, decreased tumor burden and decreased mass. And so that brings about the question of how can we effectively assess treatment efficacy and patient prognosis at early stages of ICB therapy, right? Like how can clinicians know 3 weeks in that the therapy is working? And we'll talk about this a little bit in the following slides. So, and this is important because it turns out that while this therapy has actually been very effective in a significant cohort of patients, there's a subset of tumors that are actually refractory, which means they don't respond to therapy, or the patients sort of have this initial eradication, but then a resurgence of that same tumor type with perhaps evolutionary changes. So the question really, the challenge posed to clinicians is why do these patients not respond to anti-CTLA-4 therapy in the first place? And if they do, why do some of them have relapse? And lastly, what biomarkers are predictive of successful ICB therapy, and is there something in the blood, some circulating factor in the blood that we can measure to predict, okay, this person has X level of whatever protein, we are confident that they will have a positive response to this therapy. And so there's a little bit of work being done in this regard regarding tumor antigenicity and tumor-associated antigens. So we're going to go back to the CTLA-4 example to explore this concept. So tumor-associated antigens are sort of loosely defined, but they're being explored as a prognostic marker of immune checkpoint blockade therapy. So just a reminder, we have CD28, and then we have CTLA-4 competing with CD28, and also directly inhibiting the activation of the naive Cda T cell. And then we have immune checkpoint blockades, such as ipilimumab, that act at this junction and prevent CTLA-4 from binding to CD80, 86. But we can't forget that, the original, I guess, event that spurred this CD8 activation was this antigen interaction, right? None of this CD28 stuff would matter if we didn't have antigen. And so this is where cancers really prove how smart they are, because it turns out there are variable levels of tumor antigenicity. And I'll sort of explain what I mean in a somewhat unrealistic but easier to explain, like, dichotomous example. So think of it as there are hot tumors and there are cold tumors. Hot tumors are highly immunogenic. They have tons of antigen. They are typically very painful for the patient because they are very effective at activating the immune system and causing an inflammatory response and the release of tumor necrosis factor alpha. And so even though they might be more painful in the interim, patients are better at clearing them, at least with their own innate immune system. I shouldn't say innate, intrinsic immune system. And then it would make sense that because those tumors are better at activating T cells, amplification of the immune axis would amplify or would show greater effects in that tumor population. And this is the case, but it's not the case with these quote-unquote cold tumors. So cold tumors are poorly immunogenic. They have very little antigen. And so these almost go undetected, relatively undetected by the immune system. And you'll find that there are a lot of T regulatory cells and tumor-associated macrophages that have been recruited to these cold tumors, such that even the small subset of immune cells that do react to this tumor are immediately suppressed by immune checkpoints. And so measuring a patient's tumor antigen level prior to the start of checkpoint blockade therapy is being explored. And I think some studies have come out showing that highly immunogenic cancers or tumors, patients with highly immunogenic tumors respond better to therapies, which is great because it aligns with common sense. Cool. So now that we have an understanding of tumor antigenicity as well as the godfather of checkpoints, which is CTLA-4, we're going to move into PD-1, which is actually receiving a lot more attention these days than CTLA-4. I think there are 3 or 4 approved anti-PD-1 antibodies. Anyways, the important things with PD-1 is that it actually acts at a different part of the CD8 T cell life cycle than CTLA-4. So if we remember with CTLA-4, that acted on naive Cda T cells before they differentiated. Now, when we have a differentiated Cda T cell and it's become a T effector cell, it starts expressing PD-1 on its surface. And so what happens with PD-1 is once PD-L1, which comes from various sources that we'll discuss later, once PD-L1 binds to PD-1, it causes intracellular signaling, again, glossing over some details, but that leads to either one of or both anergy and or apoptosis. So we know what apoptosis is. Anergy is just a state of exhaustion where the T cell is no longer active, it's no longer cytotoxic, it can no longer respond to foreign threats. And essentially, this is yikes, I don't want to say it's irreversible, but it's generally considered a terminal state. You'll have to fact check me on that one. I want to say it's generally a terminal state because the T cell models I've seen have exhausted T cells as a terminal state. But there could be studies on reversal of exhausted T cells. So regardless, so PD-1 is expressed on effector T cells, not naive T cells. And another important note is that it's also expressed on NK cells and B cells. And so the general thinking is that therapies targeting PD-1 can not only boost your T cell based immune response, but also your NK and B cell based immune response, really making it a more appealing, broad scope general immunotherapy. That's a lot of adjectives. And similar to what we saw in CTLA-4 with how it was expressed on T regulatory cells, PD-1 is also expressed on T regulatory cells, and it also causes the exact opposite effects on T regulatory cells in that it activates them. And instead of increasing immunosuppressive activity, it actually increases T regulatory cell proliferation. And so the thinking is that by blocking PD-1, we can not only prevent the exhaustion of T cells, but effector T cells, but we can also enhance anti-tumor immune response by diminishing the number of T regulatory cells and sort of suppressing the suppressor, which you'll find is a common theme with this particular type of therapy. Great, so we know what PD-1 does, but we have to understand where exactly does the ligand come from. So this ligand isn't some great evil that's out there just to suppress your immune system, right? It has some very useful intrinsic and endogenous effects. So for example, in an inflammatory response, your body needs a way to sort of curtail that inflammatory response, particularly towards the end of the response when most of the threat has been cleared. Otherwise, your body risks, the damage from prolonged inflammation. And so PD-L1 is both secreted and or expressed on the surface of certain cells like dendritic cells and T regulatory cells that can then make physical contact with effector T cells and cause that sort of anergic and exhausted state. And so your body has this intrinsic way of attenuating inflammation. But it turns out tumor cells also have the ability to express PD-L1, and it can also co-opt cells and recruit cells to its microenvironment that also express PD-L1. And so you can almost think of it as the tumor creates these landmines within itself, right? So when you have T cells that try to infiltrate into the solid tumor and start killing cells in the interior or the core of the tumor, once they encounter sort of a cell that is enriched for PD-L1, it's going to be turned off. It's going to be anergic, or it's going to apoptose. And so navigating the tumor microenvironment becomes very challenging for a T effector cell. But the fortunate news is, in addition to therapies against PD-1, therapies against PD-L1 have also been explored and shown to be efficacious and approved by the FDA. So there's the ability to target both of these. I think there are combination therapies approved for both of these L1s. But don't hold me to that. Yeah. So there are immune checkpoint blockade therapies for both. And so just talk a little bit about how exactly tumor cells end up expressing PD-L1, which is naturally not expressed on most of your body cells. It sort of adopts this phenotype by mimicking, I guess, your regulatory cells. And so the way the tumor cell acquires immune resistance is sort of interesting. And I should point out that there are two types of tumor immune resistance, and both innate and adaptive. And this is not to be confused with innate immunity and adaptive immunity, which is your body's response to foreign bodies. But this is your tumor's response to your immune system. And so innate resistance in your tumor cell consists of constitutive oncogenic signaling. So just by virtue of being a cancer and being highly proliferative, there's thought to be some oncogenic signaling that's just on and has to be on, and that increases the expression of PD-L1. And I'm almost tempted to think it's sort of a selective pressure, such that only cancers that end up producing PD-L1 can survive, immediate killing by the immune system. But that's just speculation. So that's innate resistance, just by virtue of being a cancer and having this particular suite of genes enriched. Adaptive resistance is where it's sort of leverages the natural infection-driven pathway of PD-L1 induction. If we go back to the infection or inflammation example I used earlier, when your body is sort of clearing out those infections by having an inflammatory response, one of the secreted factors is interferon gamma, which then causes PD-L1 upregulation in its surrounding cells. So tumor cells can actually gain the ability to respond to IFN or interferon gamma and begin expressing PD-L1 in response to inflammatory signals. And so the interesting part of that is it sort of implies that tumor cells have this innate ability to conduct immunosurveillance and be acutely aware of their surroundings and what immune cells are in the vicinity, and upregulate the corresponding immunosuppressive ligands, which is both fascinating and really troubling. And so this particular ability of the tumor cell to adapt its expression patterns has actually been shown at a surprisingly high resolution, right? Where adaptive resistance and PD-L1 expression can vary with both intratumoral lymphocyte density and interferon gamma concentration. So you would imagine that within like a tumor or a glob or something, you have tumor infiltrating lymphocytes or T cells that infiltrate the tumor heterogenously. And it's been shown that where you have higher density of T cells in a tumor, you have higher PD-L1 expression. And the same goes for interferon gamma, where there's higher concentrations of interferon gamma within a tumor, you have higher PD-L1 expression. And this sort of implies that the T cell or the tumor is dynamically and specifically responding to that immune threat, this sort of existential threat, to a high spatial temporal degree. That's not a good way to say it. With high spatial temporal granularity. So that really covers what I had to say about PD-1 and PD-L1. Now, if we just circle way back to CTLA-4, I just wanted to tie everything together so we have a better picture of where these checkpoints lie. So just to remind us, CTLA-4 signaling suppresses naive Cda T cells and prevents them from activating to then go on to proliferate and or differentiate. And this sort of occurs very early on in the life cycle of Cda T cells, whereas PD-L1 is a checkpoint that happens or is expressed on effector T cells that have already been differentiated, and sort of prevents them from having cytotoxic effects by causing their apoptosis or anarchy. And so it's sort of useful to think about these as acting on different T cell mechanisms, right? Where CTLA-4 blocks the activation of T cells and PD-1 blocks the activity of T cells. And just to really briefly touch on some of the other immune checkpoints that were mentioned in the paper, these have no FDA-approved therapies that I'm aware of, but some of these, particularly LAG-3, have extensive preclinical work done on them. So I'm just going to touch on them briefly. So LAG-3 is expressed on T regulatory cells and macrophages, dendritic cells, and effector T cells. Its ligand is MHC2. And so signaling through this, similar to PD-1 signaling, is that it boosts T regulatory cell activity and induces effector T cell anarchy. And it's interesting because dual blockade of PD-1 and LAG-3 has been shown to synergistically reverse effector T cell anarchy. Oh, I guess it's not a terminal state. I take back what I said on like slide 11. Blockade of either one of these separately is not as effective as a blockade of both of them when it comes to preventing anarchy of T effector cells, which is important because it implies they can be combined in the clinic. Unfortunately, however, in mouse models where both of these are knocked out, all of the mice pretty much develop lethal autoimmune disorders, which really just goes on to highlight the sort of narrow therapeutic window of these therapies and also how critical they are in preventing alloreactivity and autoimmune syndromes within our own bodies. Some of the other ones, that we're going to talk about in much less detail are TIM3 and galectin. So TIM3 is a receptor, galectin-9 is the ligand, where TIM3 signaling inhibits Th1 anti-tumor response. So Th1, if we recall, is derived from the CD4 plus T cell lineage. It's associated with, among T helper cells, is associated with the highest pro-inflammatory response, is how I would put it. And there's been studies showing that the dual blockade of PD-1 and TIM3 increase T cell proliferation and cytokine production. A2AR, which is an adenosine adrenergic receptor. I actually want to talk a little bit about this one because it sort of ties into the cancer metabolomics, where the ligand for adenosine adrenergic receptor is adenosine. Dying cells release adenosine, which is particularly important within tumors because, as we know, cancer cells are highly proliferative, but it turns out that there's also extremely high turnover within tumors, right? It's been shown that up to 90% of cancer mitoses are actually unsuccessful, where the mitotic cells end up dying. And it's only by mere virtue of how frequently they're undergoing cell division that the tumor is able to grow. Right, so you have massive growth, and 90% of the cells die, but you're still seeing rapid tumor growth. And so you'd imagine all these dying cells release just unimaginable amount of adenosine. And this adenosine-driven signaling through A2AR is actually a major driver of T regulatory cell differentiation. And it'll actually bias naive CD4 T cells into the T regulatory phenotype. And then the T regulatory cells, being the bad actors they are, actually continue to go on to convert AMP into additional adenosine. And this sort of feeds into this vicious positive feedback cycle. Yeah, we all know what that is. We're biologists. So adenosine released by dying cells as a big player, especially in solid tumors, where you have high cell density. And lastly, we have BTLA-4, which is similar to CTLA-4, but it's a B cell associated, and we are not going to talk about that because it is really complex, and there's a handful of others. Great, I think this might be my last slide. I just wanted to talk about some of the approved immune checkpoint blockade therapies that are on the market, because I guess it's of some personal significance and interest, because I'd like to go into immune oncology. So for CTLA-4, some antibodies against CTLA-4 have been developed. Ipilimumab by Bristol Myers Squibb. For PD-1, there's nivolumab, again by BMS in 2014, pembrolizumab by Merck, and cemiplimab by Sanofi. And I did want to talk a little bit about pembrolizumab. Brand name is Keytruda, and some of us might have seen the commercials. I bring it up because it's considered one of the most landmark, blockbuster drugs in the past decade. And once certain major patents expire, it's on track to become the best-selling drug of all time. Oh, no, that's a lie. Best-selling drug of the year. And the reason it's so widely popular is that it's approved for over 20 indications. So over 20 different types of cancer or subtypes of cancer, which is actually just a really large number. Many medications are lucky to be approved for one indication, or more than one indication, I should say. So yeah, pembrolizumab, it's a big deal, and Merck owns it. And then some PD-L1 therapies that have been developed. You have atezolizumab by Genentech, durvalumab by AstraZeneca, and arvelimab by Merck and Pfizer. So there are therapies on the market for these. And then I guess the frontier in terms of how clinicians are using these is deciding the best combination, but also the best dosing regimen. Because as we recall, there's sort of this long, several month delay between dosing and tumor shrinkage. And so they're rather complicated to study. They're rather time-consuming and economically stressful to study. and also no one likes T cells because they're so freaking complicated. So that's one frontier. Just deciding combination therapies and the right dosing regimens for these immune checkpoint blockades. And there's also some work being done to explore them in a neoadjuvant setting. So typically these are administered in the adjuvant setting, so adjuvanted surgery. So surgery to remove any tumor you can, and then you get additional chemotherapy or immunotherapy to your body's response to clear out any remaining tumor. But work is being done to actually reverse the order of things and have immunotherapy be done first to sort of shrink the tumor and make it easier to clear by surgery. So work in that field is ongoing. And the immune system is complex, and I hope this made sense because that's all I had.